It is a common misconception that transfection requires plasmids purified with endotoxin-free kits. The scientific reality is that only dramatically high levels of endotoxin, above 100 EU per microgram of plasmid DNA, have been shown to negatively impact transfection and protein expression in standard cell lines. The very low endotoxin levels of 0.1 to 1.0 EU per microgram produced in anion exchange plasmid purification systems, like our PureLink HiPure kits, are ideal for the majority of transfection applications. To get started, prep the column by adding 30 milliliters of equilibration buffer to the combined filter and DNA binding column. Let it empty by gravity flow while you prepare the bacterial pellet. After spinning down your cells into a pellet and removing the media, add 10 milliliters of resuspension buffer. Pipette up and down until there are no visible clumps. Next, add 10 milliliters of lysis buffer and mix by inverting the tube 4 to 6 times. Leave at room temperature for 5 minutes. Then add 10 milliliters of precipitation buffer. Mix again by inverting the tube. Immediately add your samples to the pre-equilibrated filter DNA binding column. Allow it to empty by gravity flow into a waste receptacle. The clarification filter eliminates an additional centrifugation step and the need for hard-to-use plunger-based purification systems, allowing for parallel processing of plasmid DNA purifications. After the lysate has been clarified and passed through the filter, remove the clarification filter and discard. Your plasmid DNA is now on the column. Now, wash your plasmid DNA by adding 50 milliliters of wash buffer to the side of the column. Let it empty by gravity flow. Place a clean 30 ml round bottom tube or similar under the column and add 15 milliliters of elution buffer. Let it empty by gravity flow. Your plasmid DNA is now in the tube. Add 10.5 milliliters of room temperature isopropanol to your plasmid. Mix gently by inverting. And centrifuge at 12,000 times G for 30 minutes at 4 degrees Celsius. Gently pour off the isopropanol, ensuring you can see the plasmid DNA pellet. Carefully add 5 milliliters of 70% ethanol and centrifuge at 12,000 times G for 5 minutes at 4 degrees Celsius. Decant the ethanol and place the tube upside down on a lab-grade tissue to air dry for 10 minutes. Resuspend the pellet in TE. Your transfection-grade plasmid DNA is ready to be used or stored at -20 degrees Celsius.